The purpose of this presentation is to describe the WHO project and where it currently stands. The project started 2 years ago with the goal of profiling every single known blood cancer defined by the World Health Organization. The idea is to profile over 10,000 cases by DNA and RNA sequencing and to represent every single known hematologic malignancy. Importantly, the individual entities in the blood cancers book serve as projects that can be led by each participating group. The idea is that every institution provides cases across the entire spectrum of WHO entities, so over 100 plus blood cancers, and individual projects will use cases from the entire group. Our group will provide whole exome and transcriptome sequencing results and assist with the generation of the paper. But authorship is defined by each participating group that specifies which entity they are interested in. So for example, if a particular group has picked primary diffuse large cell lymphoma of the central nervous system, then all the cases that are collected from all institutions will be profiled by our group, but the data will be made available to the group doing the CNS lymphoma study, and they will own the rights to authorship and the results, with the idea that they will be inclusive of everyone who provided cases. These 20 plus papers will be followed by one paper at the end that is delayed by about 6 months that will include all the 10,000 plus cases. This is where things stand currently. Sample collection is nearly complete with over 11,000 samples enrolled in our sample collection system. We will continue to collect samples through summer of 2019, including new collaborators where people want to participate and are able. We also have a continuing need for rare entities that we will continue to collect through summer of 2019. This project is enabled by new assays and analysis. First, the assay. The blue box shows the assay that we used to perform for next generation sequencing. This was, for example, the assay that we used for our 1,000 diffuse large cell lymphoma project. And the workflow is separate for DNA and RNA and spans about 2 days of a technician's time. We have greatly simplified this process. So the whole thing start to finish takes a little less than 8 hours. So you go from sample to complete DNA and RNA sequencing library in the span of a single technician's workday. What we measure is both DNA and RNA. That includes all genes. So this is whole exome level sequencing, plus all oncogenic viruses. So Epstein-Barr virus, Hep B, Hep C, and so on. The entire immunoglobulin and T cell receptor locus, non-coding genes like lincRNAs, microRNAs, and so on. All known major fusion and translocation breakpoints, promoter, enhancer, and untranslated regions for all cancer genes. And importantly, we embed controls at every stage of the assay that enables really finely calibrated measurement of both expression and DNA copy number. This slide shows how being able to finely calibrate expression is a critical feature of our assay. Most gene expression varies continuously. So you can see this in the case of BCL2, where expression goes from dark blue to dark red without a clear cut point which distinguishes really high from really low samples. That distinction is really arbitrary. Having our embedded controls allows us to finely measure the distinctions between small differences in expression. So sample 1 versus sample 2 in this case. And that allows us to place new samples like sample X accurately within that spectrum. And so having these controls in place allows us to place new samples in that spectrum and enables accurate comparisons within and across diagnoses. So now you can compare BCL2 expression in follicular lymphoma compared to diffuse large cell lymphoma compared to even AML, because everything is run on exactly the same platform with exactly the same controls. The other major innovation in our work is in terms of the development in bioinformatics. The old approach for bioinformatics used separate analytical programs for calling mutation, copy number, expression, and so on. This used to be a very labor-intensive task for bioinformaticians and required constant management of infrastructure. We have now condensed that into a cloud-based automated approach that takes the data as it comes off the sequencer and automatically triggers the RNA-seq and DNA sequencing analytical pipelines. These are directed by a program that we call Cloud Conductor, which communicates clearly through the sample database and generates results. And this entire pipeline automates a lot of the early tasks that took up a lot of time for our bioinformaticians and automates the management of infrastructure in a way that was just not even possible 2 years ago. All the samples are being subjected to pathology review, which is being led by Dr. Naresh Kekeri at Imperial College London. As most people using the system are well aware, we have a web-based system that collects both pathology reports and sample images. The idea is to provide just enough information that another pathologist can make a determination of the diagnosis. So information in the pathology report counts as a performed assay, and Naresh has been directing this process, and the idea is to start with T-cell lymphomas, which are somewhat difficult to diagnose, but there are fewer cases. This is being tested right now and will be rolled out starting in December of 2018, with the idea of being complete by June of 2019. Clinical data collection is being led by Jean Koff out of Emory University. We have designed a number of clinical data templates which have been circulated through the group. We ask that people provide an H&P and oncology note from the last follow-up. Our clinical coordinator will provide redaction and de-identification. Where possible, we like to collect dates, but this is not always feasible, and some institutions forbid this. And we have an on-screen calculator that allows people to put in dates and save their age at different time points without you having to do all the heavy lifting. We have separate templates for each disease with some disease-specific templates. Shown here is a diffuse large cell lymphoma template. Other templates for acute myeloid leukemia, for example, and these are automatically loaded up for each sample based on its diagnosis. There are two projects that require actual survival data. These are diffuse large cell lymphoma and follicular lymphoma, for which we'll be collecting 1,000 cases each. And this is an opportunity for technology development and new application. The idea is that these papers won't just include these cases, they would also include other elements such as single cell sequencing. One example is shown here, where we can perform RNA-seq on thousands of cells from a single biopsy and distinguish different cells in the tumor microenvironment. Another aspect that we will build in is mouse models of selected mutations. And so these projects still need cases, which we will continue to collect through summer of next year. So this is the summary of clinical data collection. The clinicians are testing and finalizing data collection system in December, and the idea is to start collecting data in January with the goal of completing it by summer of 2019. This is a very time-consuming effort, and it is also a really critical element of our sample and data collection. everything that comes after this depends heavily on getting these details correct, and we will do everything we can to get it right. I'm going to now talk about the WHO workshop, which is planned for sometime in 2019. The goals of the workshop will be to share data and provide a common base of knowledge from the data that we have generated from the 10,000 plus samples. The immediate goal will be to prepare and submit an abstract to the American Society of Hematology or the USCAP meetings. And we'll develop a framework for each paper so that each group that is participating will leave with an outline and early draft of figures. The timing is still to be arranged. I will circulate dates for June and July in the weeks to come. Once we agree upon a set of dates, it will enable people to block that time off and plan their clinical time and vacation accordingly. The general structure of the workshop will include a large group session around a particular concept, which, for example, mutation calling or DNA sequencing, where we'll talk about the general concepts, and that'll be followed by a small group breakout session that'll go over project-specific data. So here, each institution will have access to the data for their project and will understand how this fits with the big picture of analysis and so on. There will also be panels and discussion around larger issues related to next-generation sequencing, analysis, and so on, as well as future directions of the project. For each individual project, a bioinformatician will be assigned for long-term follow-up, and this individual will also schedule bi-weekly office hours that enables groups to follow up and ask questions. The idea is to provide support throughout the publication process that goes beyond the workshop. This is just a preliminary agenda, but it shows you kind of how we have structured this and thought about this. The sessions are structured around the individual things that we're measuring: DNA, RNA, clinical pathological correlations, and there will also be panels that discuss translation and pathology and clinical trials, as well as thinking about future studies and governance around our. There's a lot that can be done with this data and the approaches that we have developed for this project. There are ideas already being put forth and for clinical trials and follow-up projects. And having a clinical assay greatly enhances our ability to do other projects. So I don't think about this, or at least I try not to think about this too much right now, but there is clearly many productive directions that we can take this data and assay in. One immediate project is a parallel project in Africa, where individuals and institutions in Africa provide samples in a very parallel fashion that uses our systems and assays and generates a comprehensive spectrum of different cancers in Africa, this time led by individuals in Africa. And so if you have connections or an interest in participating in some of these projects, please send me an email and we can follow up. This is our last slide. There is still a lot of work to do in terms of pathology review, clinical data collection, as well as generating the sequencing data and analysis. This is a foundation for a lot of fun projects ahead, even beyond the immediate WHO project. I want to thank you sincerely for all the hard work you've done and look forward to working with you through the WHO workshop and the years to come. Please email me if you have any other questions or concerns. We'll be talking soon. Thanks.